Generally, when people think about cyanobacteria, it's often in the news for negative reasons. It's clogging up a lake somewhere. There's a lock in Northern Ireland where it seems to do that quite a lot. But actually, the things that people don't realize about cyanobacteria are they are the most efficient photosynthetic organisms on the planet. They are the reason our planet developed a breathable atmosphere in the first place. So that was our starting point. We thought, well, that's got great potential. And then you look at the similarities with other microbes, E. coli, streptococcus, yeast, so on and so forth, that have been engineered to make useful products. And we think, well, can cyanobacteria do that? And the more we looked at it, the more we realized cyanobacteria could do that for a whole swathe of chemicals.
What if the ocean held the key to decarbonizing the entire chemicals industry? Today's guest spent 3 decades in commercial and leadership roles before discovering a remarkable microbe floating off Singapore's coast. Cyanobacteria that could revolutionize how we manufacture everything from cosmetics to fuels. Tim Corcoran, who is the CEO of Deep Blue Biotech, joins us to reveal how this marine organism secretes high-value molecules directly into seawater. Why photosynthesis changes manufacturing economics, and what finally makes cyanobacteria commercially viable after years of failed attempts. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brolman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Tim. It's good to have you on today.
Thank you for having me.
It's a pleasure, Tim. Share something that you believe about bioprocess development that most people disagree with.
Oh, goodness. I think a lot of people view scaling bioprocesses as inherently difficult, quite off-putting. I don't think it necessarily needs to be the case. I think the technology is improving. People are learning at a rate of knots. We personally have designed our technology specifically with scaling in mind to make it as easy and as straightforward as possible. But certainly when you speak to people, that is often one of the primary concerns on their minds.
Take us back into your story before we talk about the exciting science and technology you're developing. Tell us what sparked your interest in commercial and leadership. You're coming from a different background, and tell us also what led you to science and finally the role you're currently in.
I started off, I think when I was about 16 with my first sales job, just a summer job selling double glazing, which does not have a great reputation here in the UK, and it may well be the case elsewhere. But I realized that I quite enjoyed it and that I was competent. So when I left university, I didn't know what to do with my economic history degree. So I thought, well, I've been doing this through university to pay some bills. I'll carry on doing it. And then over the years, that developed from a sales into a broader commercial and operations role. I enjoyed looking at how businesses could commercialize, how they could grow revenues, how they could improve profits. For a good number of years, I worked for a market research company which was dealing with innovation, specifically within the FMCG market. And they were looking at what successful innovations did as case studies and why some innovations failed. And I always found that very thought-provoking to look at. There were certain traits you saw time and time again in successful innovations. And so that informed a lot of my thinking about product development and about how you could create new things that people actually wanted and which would succeed. I then became a business development consultant and I was working with a lot of early stage companies, helping them take what was often just an idea on a piece of paper and turn it into a reality, helping them with investment, helping them with planning and strategy and commercialization. In particular, I was running a company called Master Investor at the time, working with a chap called Jim Mellon, who's a fairly visionary investor chap. He's very, very keen on science in particular and how it can fix a lot of our problems and how you can use that to make money at the same time. And in particular, he was always very keen on things like alternative proteins. He had a company called Agronomics, still does, who have invested heavily in the sort of the alternative proteins market. And so I was getting through him exposed to a lot of really interesting companies. And it was that gradual process of thinking about innovation, thinking about how you commercialize things, talking about people who are frankly brilliant at what they did and learning from them about how they could transform that into a living, breathing, hopefully profitable company.
What was that pivotal moment then that made you take the leap and found Deep Blue Biotech? And what was the vision behind?
It'd been percolating away at the back of my mind for a while. I'd been working with these companies and I knew I'd been able to help them. And I thought, well, why don't I try and do it for myself? At the same time, I have a long-standing and deep concern about climate change. I'd wanted to do something about it and I wasn't sure how I could do something about it. I thought, well, if I help to commercialize climate-friendly technologies, then potentially that means I can sleep at night. I feel as though I've made a positive difference. So I joined an accelerator or a venture builder program, to be more precise, run by an organization called Carbon 13. So they're trying to create climate-focused companies, and they bring together technical people, scientists and engineers and things like that, and commercial people, people like me, put them essentially in a room together for a 3-month period and see what comes out. So I met my co-founder there. He's a chemical engineer by training. He'd been working as VP of Sustainable Innovation at Unilever, and he'd grown increasingly frustrated because all the sustainable ingredients and chemicals that were brought to him were either too expensive or they weren't as good as the ingredients they were already using. And Unilever weren't going to go for that, and consumers weren't going to go for that. They didn't want to pay more, and they certainly didn't want something which wasn't effective. So he'd left looking for a means to create a technology that could do that. And over that process of 3 months at Carbon 13, we looked at different technological options and we explored it, and we eventually settled on cyanobacteria. And the more we looked at it, the more we thought this has got huge potential.
That sounds exciting. And tell us more about these cyanobacteria and why this is an interesting host to work with.
I have a tendency to get overexcited at this point, so I'll try to keep myself down. Generally, when people think about cyanobacteria, it's often in the news for negative reasons. It's clogging up a lake somewhere. There's a lock in Northern Ireland where it seems to do that quite a lot. But actually, the things that people don't realize about cyanobacteria are they are the most efficient photosynthetic organisms on the planet. They are the reason our planet developed a breathable atmosphere in the first place. So that was our starting point. We thought, well, that's got great potential. And then you look at the similarities with other microbes, E. coli, streptococcus, yeast, so on and so forth, that have been engineered to make useful product. And we think, well, can cyanobacteria do that? And the more we looked at it, the more we realized cyanobacteria could do that for a whole swathe of chemicals. Now, traditionally, people have been held back with cyanobacteria because it grows more slowly than some of these other microbes, and the yields were often quite disappointing. So it was hard to commercialize, but also there was a relative lack of scientific knowledge about cyanobacteria, the tools for genetically modifying it, the understanding of how to cultivate it efficiently. That has been developing over the last few years. And then the strain that we're working with was discovered about 5 years ago now, I think. So it's a relatively recently discovered strain, not well known, which does throw up problems when you're trying to do things with it because you're learning things for the first time that no one else has had to go through. But equally, it has huge potential because it grows much faster than other cyanobacteria strains. It achieves quite high biomass rates, and it's been proven to yield commercially viable amounts of chemicals and ingredients. So when you take that as your base chassis and you then think, okay, how can I improve it? It holds enormous potential to finally realize what cyanobacteria can really do.
How is that vision linked to the discovery of cyanobacteria? Tell us more about that.
Our strain of cyanobacteria is an ocean-based strain. Now, that's sort of important because it means it tends to be more robust. It's used to dealing with a range of different sort of light and temperature, CO2 and other ingredients in the ocean. So because it's robust, it means it can stand up to changes in its environment a lot better. And potentially, and we're working on this at the moment, you can tweak its environment and eventually get to a point where its production and its growth is optimized. Now, the fact that it's an ocean-based strain means that the chemicals it produces can be described as ocean-derived. Now, in certain industries, that doesn't matter so much, but in personal care, which is the industry we're working in at the moment, that really matters. Consumers like ocean-derived ingredients. They will pay more for them. They will choose them over other types of ingredients. There is a positive effect there. Now, our first product, hyaluronic acid, our hyaluronic acid will be the only ocean-derived hyaluronic acid in the market. And so when you talk to personal care companies about this, and we've spoken to a lot of them, that's the point at which I think they start to see dollar signs flashing past their eyes, because they can see a way to create unique products with unique marketing claims, which will drive price premium, it will grow sales, it will improve their profits. Now, at the same time, we're able to say, well, look, the feedstock here is CO2. So it's carbon neutral. It is potentially carbon negative if we choose our electricity sources quite carefully. And because it's such a simple mechanism, it's a very clean, efficient process, we can make these ingredients for less than you're currently paying. So that green premium I mentioned earlier, that's no longer a factor. Worries about whether or not it's effective or not are no longer a factor because it's a drop-in solution. And you've got these unique marketing claims around the ocean-derived side. And it creates quite a compelling proposition.
Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. You definitely got a lot of unique selling points going for you. The net zero, the ocean-derived. It's pretty good. Help people better understand the differences between cyanobacteria and some more legacy cells. Gets really quickly confusing.
It does.
We have E. coli, we have moss, we have microalgae. What are the main differences?
I guess the starting point is prokaryotes, which includes E. coli and cyanobacteria, which have no nucleus. Eukaryotes, which are things like microalgae and pretty much everything upwards. Eukaryotes are fundamentally more complex organisms. Prokaryotes are simple, and from a genetic editing point of view, that's advantageous. It also means they're quite efficient. So from a cyanobacteria point of view versus microalgae, for example, which are essentially very small single cellular plants, cyanobacteria are simpler. And that means the photosynthetic process tends to be more efficient. Because the photosynthetic process is more efficient, that conversion of CO2 into chemicals is more efficient. So that's sort of where it lies. Other prokaryotes like E. coli and things like that, they're not photosynthetic. Cyanobacteria are somewhat unique in being essentially photosynthetic bacteria. So they sit almost in between the two. And arguably you could create a whole separate sort of classification for them.
Besides the photosynthesis and the CO2 aspects, are there some other advantages and work with cyanobacteria?
Because the inputs are so limited, you're not feeding it sugar. So take E. coli as an example. It generally wants sugar-based feedstocks. Cyanobacteria, the feedstock is CO2. The energy source for cyanobacteria is light, which means you can potentially capitalize on natural sunlight. So that again reduces your CO2 footprint and it reduces your costs. And then the other key aspect is the media in which it exists, and the resultant media, once it's finished doing what it does, is very simple. So in contrast to things like Streptococcus and E. coli, there are no endotoxins involved. The media is essentially a combination of polysaccharides, water and salt. And that means the downstream processing is incredibly efficient. And that's one of the things that really drives our cost of production down and makes us hugely competitive against existing methods.
So this leads me to this question then. What I'm hearing, Tim, is that the current strain has a lot of advantages and there's a lot of potential. But why is it that not many people have worked with cyanobacteria yet? And it seems to be that nowadays several companies either started to see the potential or there seems to be another factor going. Why is it now?
People have been trying to make this into an industrially viable sort of microbe or technology, to describe it, for well over a decade. And most cyanobacteria, the growth rate is multiples and multiples slower than other microbes. And so that's a challenge to begin with. And then the yields, you're looking at tiny, tiny fractions. Even after the genetic engineering, the yields would be tiny fractions of what you might get from E. coli or streptococcus or, pardon me, or yeast or something. So that was always what put people off. And then, as I mentioned, the recently discovered strain, that was one of the triggers because suddenly it's not as fast as these other microbes, but the gap is substantially reduced, which enables greater efficiency. But also the tools for cyanobacteria and the understanding of cyanobacteria's genome, they have been developed. So we're working with people like Alistair McCormick at the University of Edinburgh, who has developed a lot of the tools for efficient genetic modification of cyanobacteria and of our strain in particular. So that makes the research and development process simpler. I don't want to say it's easy because it's not, but it makes it feasible within, you look at the cost that you put into an R&D process, it makes it achievable within reasonable amounts of money and reasonable amounts of time.
One advantage I see is that your product is directly secreted versus when you're working with E. coli, you have to break up the cells first and so on. I think this can definitely offset the slower growth rates, for instance, or even the lower yield.
Definitely. I mean, having it secreted into the media simplifies that downstream process substantially. We were looking at the cost of production, and we think that reduces the cost of production by anything between a quarter and a third, which is substantial. So it makes a massive difference. The steps, and it also makes a difference from the energetic usage point of view because there's fewer steps and they're simpler steps and they're less energetic steps. So the CO2 footprint of what you're doing is reduced as well.
And how is the scale up? Because you need light, you need CO2, the right temperature. How easy? And you said you built in the scale up right from the beginning.
One of the things that we liked about cyanobacteria is that they grow in typically things like photobioreactors, which are essentially a series of glass tubes. They're modular by nature, and that means you don't see a significant difference in performance between, say, 100 or 1,000 litres or 10,000 litres because you're just adding more glass tubes. It's more a case of scaling out than it is scaling up. Now, I don't want to minimize the challenges involved. There will always be some challenges. So as you scale up, you have to think about access to light. You've got to make sure all of the microbes are getting access to the light coming in. You've got to make sure that the temperature remains roughly consistent because, again, the more light, generally, the more heat you get with it. So you've got to try and keep that controlled. And you've also got to think about the CO2 mixing, again, making sure all of the cyanobacteria are getting equal access. But actually, there is enormous headroom on photobioreactor development. There's some really interesting companies coming up with some unique models to tackle these things and make it more repeatable and scalable. Companies like Algeny, for example, in Australia, who are developing a helical photobioreactor for continuous production. It makes the entire process substantially more efficient. Now, we've actually not factored in these yet. We're hoping to do some testing with these new photobioreactors in the future, where potentially our cost of production comes down even further because of them. In contrast to bioreactors, I think people have been investing in and developing bioreactor technology and infrastructure for a long time now, and that reached a fairly sophisticated level. I think photobioreactors will take a similar path, but they're probably 10, 20 years behind. So the headroom for improvement is really quite exciting.
Now, let's talk about the business side of things. And since you have a commercial background, I'm very curious about your thinking behind what kinds of products you have chosen. And tell us, what were the reasons behind the choices and also the business choices you made?
It's a good question. So at the time that we formed in 2023, it was around the time that money was getting quite expensive. Interest rates were going up. People weren't investing and lending money quite like they had done for the previous 10 or 20 years. So we consciously thought about, well, how can we be profitable quickly so that we don't have to keep going back to the well? I think the days when synthetic biology companies take 10 years to develop a product that was commercially viable are gone. You won't get the time for that. Well, the very first piece of research we did was, what is our cyanobacteria predisposed to make? What chemical precursors does it contain? And there's a really long list. So that was a good starting point. It did then make a lot of work for us, though, because what we had to do is take that list and combine it with market sizes and market prices, because we wanted something with a large market and ideally a high price. If it's got a high price, then we could be competitive quickly. So when you take that Venn diagram and you sort of transpose all of those different factors, there were a number of candidates in there, but the single best candidate to start with was hyaluronic acid, because it's expensive. You're generally looking $2,000 per kilogram, often sometimes quite a bit more than that. The market is big and it's growing quickly. But also the other factor was hyaluronic acid is very popular in the personal care sector, where storytelling matters. Now, you can use it in pharmaceuticals, for example, and sort of therapeutics, but the storytelling matters a lot less there. If you're in that sector, people just want it to work. Whereas in personal care, when you talk about the origins of the ingredient, you talk about ocean-derived, you talk about carbon neutral, that matters, that adds value to the product. People will pay to a greater or lesser extent for that. The other factor that we like about the personal care sector is, from a regulatory point of view, it's much more accessible. If you want to get into pharma or food, the regulatory barriers are somewhat intimidating. They are time consuming and they're expensive to deal with. Personal care, obviously there are regulatory barriers, particularly safety and efficacy testing and things like that. But relatively speaking, it's a shorter, less expensive process, and there is a well-trodden path for biotech type solutions in personal care products. So all of that together, it meant that we ended up with a nigh on, as far as we were concerned, a more or less perfect business case for hyaluronic acid. We do have a list of second, third, and fourth generation products that we're going to tap into once we've got the hyaluronic acid up and running and profitable. But also, as the technology improves and becomes more efficient, you can move down the value chain and tackle slightly less expensive products until hopefully you get towards the sort of the commodity end of the market. Because from an environmental perspective, that's where you'll have the biggest impact.
Your approach sounds a lot like the Tesla model, where you start with the premium product to earn some money quickly, and then you walk down, should I say, the value chain, or at least the cost. This is an important message. I just want to stress that again, because a lot of people listening are scientists, and we think about the science. Tim, you have a commercial background, so tell the scientists listening why your approach is such a game changer. What changes when you start with the premium product first?
When you start with a premium product, it means the technology doesn't have to be perfect to go to market. You've got a far better chance of taking something which is good enough and turning it into a profitable business. Now, in the years I worked as a business development consultant, I worked with a lot of brilliant people who were fantastic at what they did, but they didn't have that commercial expertise. They didn't know how to translate that. There are people all over the place, business development consultants like me. If you're at a university, they will have entire departments dedicated to taking technical ideas and translating those. And what I would say to any scientist with an idea out there is go and speak to these people, ask them, do you think this could work? How could it work? Because they can help you with the planning. They can help you with identifying the customers. One of the things when I was working in innovation market research a few years ago, one of the key characteristics of successful innovation, successful new products, was understanding the market, understanding what people want. Now, that's not necessarily a job of the scientist. They'll need people to help them with that. But speak to your potential customers, understand what it is they want. I think if you go back 20 years, maybe more, companies used to come up with a new product and then try and find a way of chucking it at the market and hoping it stuck. I think in the last 10 years in particular, companies have become a lot more efficient, a lot more intelligent about it. They look at the market, they look at what the market wants, where the gaps are, and then they start on the innovation process. And they end up with products which hopefully are perfectly suited to an unmet need in the market. So that market research, customer discovery is a vital part of the process. Before you start sinking significant amounts of money into sort of the development and commercialization, you need to understand that, because that can help guide your subsequent research.
And what are some commodity products you think could still be interesting, but where you have a huge ecological benefits? What are you thinking about?
From our point of view, biofuels is an area of strong interest. Now, cyanobacteria are able to make things like butanol, for example. Now, butanol currently isn't used as a biofuel because there isn't really a clean, efficient way of making it. But actually, as a biofuel, it holds great potential because it has a far closer profile to petrol and diesel than ethanol does. So if you can make butanol in a carbon neutral fashion, it has great potential as a biofuel. Now, the challenge is butanol is currently it's about $2 a litre. So it's cheap. And generally speaking, people don't want to pay a huge amount more for their fuel. So the goal is to be able to make butanol efficiently. Now, the thing that the triggers for us to be able to do that, and it's somewhere in the future, but we are going to be working on it further, is we need to get the yields from the cyanobacteria up. Now, the good news is our cyanobacteria strain, the yields that we've already got are actually looking far more promising than we ever expected. So we actually think there are multiples of what we thought were achievable are now achievable. The cost of productions are much lower than we initially expected, and the technology continues to improve. So you're going to scaling up to deal with a biofuel to make a significant impact on, the petrol market or the diesel market or something like that. That's a somewhat intimidating prospect because you think about the scale of the petrochemical industry. But if you want to replace them, you do need to be able to scale it up. And so if you want to have a photobioreactor producing butanol that isn't the size of a small country, the critical points will be that yield, getting a few grams per litre, perhaps 5 to 10 grams per litre into that photobioreactor, at which point it starts to look potentially like quite an efficient way of doing it. The environmental impact is absolutely mind-blowing.
We have explored why cyanobacteria's unique biology, photosynthesis, CO2 utilization, and direct secretion finally makes commercial sense. In part 2, we'll dive into the strategic decisions that separate successful synthetic biology from brilliant failures. We'll talk about choosing hyaluronic acid over commodity fuels, navigating photobioreactor scale-up, and building toward a licensing model instead of capital-intensive facilities. If this episode sparked insights, leave a review on Apple Podcasts or your favorite platform. All right, smart scientists, that's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.